Hi everyone, welcome to this presentation that we have today. Today we are going to be going through to have a look at how the NCBI virus website can be used to help you with your SARS-CoV-2 research. My name is Dr. Anusha and I am a scientist here at BenCos. So let's get started. So the first thing we need to do is just to look at NCBI's page. Now they have a separate page for virus. So we are just going to search for the NCBI virus database. When you Google search, this should be the first result that you might get. So currently we are at the home page of NCBI virus. If we just scroll down and have a look, there is a visual dashboard which is always present. And there are different details about NCBI and the host distribution of the viruses and so on. We won't be looking at that in detail today. Let's focus on NCBI virus for SARS-CoV-2. Right now, since we are in the middle of the pandemic, a lot of websites, including NCBI, have included quick access to data which is available for SARS-CoV-2. So we will be looking at the quick access which is available right here. Now let's just enter and have a look at the RevSec genomes. So this is a page that we will enter once we click on the RevSec genomes. So for SARS coronavirus 2, there is only one reference genome which is available, which is seen here as the RevSec genome. Apart from the reference genome, of course, there are a lot of genomes which are available for us to access, and all the genomes would be present in the tab which is called the nucleotide tab here. So there is nucleotide, there is protein as well as RevSec. So currently, let's just look at the nucleotide tab because today we are going to be focusing more on the genome of SARS-CoV-2. So once I click on the nucleotide tab, there are a number of results that would arise on the page here. Let us take a little bit of note on what are the options available to us and how we can use them for some different analysis that we might want to do. First, let's look at the tab right on top. There is an accession number. As we hover through these tabs, we will be able to see what it is specific for. So the accession number is the unique sequence ID or this unique sequence accession number, which we can note down and use for publications, and we could also use it in order to search for a particular sequence. We also have other information about the sequence, such as the release date, what species it is, the length, and so on. Apart from what are already present on the tab, we can either select or deselect based on our preference. So let me just go to the select column sections. So here we can see a number of columns which are displayed. And for example, say I don't want the species because I know that this particular data which I'm looking at contains only SARS coronavirus 2. So I don't really have to be bothered about the species. I can just remove this from the display. But say I want some other information. For example, I want information about who are the authors who have really submitted this sequence. I will just include this into the displayed column section. I might also want to include something else, like say the publications. So once I do this, I would just apply. And there are the results shown remain the same. However, there is additional information about the authors as well as the publications, which is right here. So we can scroll towards the right to have a look at the different information that is available about the genome samples that are submitted to NCBI. So currently we have nucleotides and the information that we require from the nucleotides as well. We can scroll towards the right and see what all information is available to us. So there is a lot of filtering that could take place as well, which we can use if required. Now, in this case, let me go to nucleotide completeness. What this means is whether the complete genome is present or not. If I want to look only at complete genomes, I can just help to press the upward arrow and it will show me complete genomes first, followed by partial genomes. Say I wanted the other way around, I will press on the bottom arrow and first it will start off with partial genomes. In the same way, if I want to change something based on the release date, I can again press the upward arrow and now the first releases will come on the top of my page. And if I want to do it in the other order, I will just press on the downward arrow and we can see that this particular sequence has just been released a few days ago. So this is how we can use the tab on top to change or filter for any information that is important to us and which we might want to know more about. The other thing which is very important is to look at these particular features which are present towards the left side of the page. So we can specifically ask for a particular accession number by pressing on the accession column. Now, for example, let me look at one accession number, the one which is right on top of the page. So once I submit, only that accession number is going to be displayed. And if I don't want only this one to be displayed, I can just press on the cross mark and then everything else is going to be displayed now. The other important aspect is looking at the sequence length. Most researchers know that the entire length of the reference genome was 29903. So if I want to look at sequences which are between 29,000 and 29,903, I can filter out the sequences which fall under that particular length, and we have these sequences here. One important aspect of this is to look at the geographical location. Now, for example, say I am only interested in genomes that are from India. I can just choose only these particular nucleotides. So we have 562 nucleotides all from India. And we can have a look at the authors as well as the geolocation all in one page. Another feature which I think is very interesting is to look at the isolation source. Most people have gotten these genomes from the oronasopharynx, which is why this has the most number of genomes, but it is possible to get genomes from other locations as well. And if we press on the location, we will have the samples which are only from a particular isolation source. Apart from that, we can also change our parameters on the collection date, release date, and so on. But that is something that I'm sure you can all explore by yourselves, and I'm not going to go into that into detail. So now let me just show you something else that is possible. Currently, the order that is being used is by the release date, and I want to change the order to show me by the geographical location. So when I press on this geographical location, it will filter this column or arrange this column based on the alphabetical order of which country has the samples. So as we know, A is Australia starts with an A, which is why it is the one which occurs first. And if we want to go to the 115th page, you just enter here. Right, these are empty or unknown locations. So let me just go to page number something like 90. And we see USA, Washington is on this page, right? So now I would want to do something. I would want to compare some samples which are between any three countries. So let me go to page 30, which I know contains samples from China. And I know that this is the reference genome because we can see this particular blue tab saying RefSeq here. So I want to choose that sample. And I'm going to go to a page which has information about the Indian genome. It's not on this page. So I will just choose maybe two samples from India. And let's just choose random samples from different countries to just look at what we can see. So I'm choosing a mix of complete as well as partial genomes just to have a look at how they could be different. Right. So I finish choosing some genomes that are of interest to me, and I have selected around 11 genomes. So there are a few things that we can do right on the NCBI page, which is to align them and to build a phylogenetic tree. So say I want to do an alignment, I can just press on the align and it will perform an alignment, which is a multiple alignment. So after we try to align the genomes that we looked at, we get a map which looks something like this. So when we look at this map, right here is a consensus sequence followed by the different genomes that we looked at. There is no particular order to this. And let's just go a little more into detail and see what the different symbols right here mean. So here we can see some parts are grayed and some parts are as a red color bar which is present. So one thing we would need to do is to zoom in a little bit, and the zoom option is present right on top. So I want to move. So the arrows here can be also used to move around the genome. And I want to zoom in to look at these two mutations which are present. And I can see this particular mutation. So what consensus means is that all these particular genomes which have a dot right here, all these represent a G. That is why it is a consensus that at this particular position there is a G. When there is something highlighted in red, it means that for this sequence, instead of a G, there is a T at this particular position. And as you might have observed, as we hover over this genome, there is some information about the genome and about the alignment position. And that can change depending on which genome we are looking at. So just click outside and then hover around a different genome. So this is something interesting if we want to take a look at the genomes just as an overall picture. So when we zoom out, we would be able to see regions which are grayed out. So if you look at regions here which are in darker gray, something like the ones here, those are the regions where the sequencing has not worked too well, and therefore there are a number of ends. So there has not been an assigned nucleotide at that position, because of which there is a gray. So let's try to zoom in and see whether we can observe that as well. So the arrows here, which are for pan left and pan right, are given for us to have the effect of moving through the genome. Right, so this is what I wanted to demonstrate. Now we can see that this particular genome has a lot of ends at this position, therefore it hasn't been sequenced properly, probably. So that is something interesting. And the other interesting thing is when we press these arrows right here. So when I click this, there is an option of expand. So what is observed here is that there is a Q, W, M, V. So this we know are the amino acids. So that gives the effect that we can even observe amino acid changes using this particular alignment option on the NCBI virus. So for example, if I want to look at the reference genome and look at another genome, I could even look at it in this way and have a look at whether there is the same amino acid encoded for a particular region. And if I want to pan through the genome, of course, this is a very tiresome way, but this is something that is possible and we can do this. So these are the different options present for multiple alignment. Now let's look at what are the options or what happens when we press on the phylogenetic tree. Now again, we have selected the same number of genomes and we will go to build a phylogenetic tree. And right here we have a phylogenetic tree, which is already represented for us. So we can look at the first genome here, which is from China, which incidentally is the reference genome. And we see that all the other genomes seem to have come from the reference genome. And we see that the genomes, which two of the genomes from Iran and one genome from India, seems to be phylogenetically quite far away from the genome or the reference genome, which was from China. Again, there are options in this particular phylogenetic tree as well. Now, we only have about 11 genomes, because of which it's not so difficult for us to look for a genome. But if there are many, we can use this find option. And it helps us to find the genome that we are looking for. We can also change the layout of the phylogenetic tree. Currently, it is a rectangular cladogram. We can also change it to a radial tree, where the diagram looks somewhat like this. Or we could change it to something like a circular tree, where this is the particular format used. We could also edit labels if required. Now, currently, I wouldn't want to do it, so this is something that can be explored as well. Reset the selections that I had already done, and I want to select any one genome which I want to download. So there is a download option right here, and there are different formats that can be used to download the data. We could use the FASTA format with nucleotides and the coding region or the coding region or the protein. Let's just try the simplest FASTA format. Next. And we are only looking to download the selected record because we just want to download one record. Particular details such as accession lists or even what is available for us to view on the table. So I just want to download that information, and I will say I want to download all the records. Here I can select whatever columns that I require. Say I want to select sequence type and genotype, but I don't want to know the host and I don't want to know the isolation source. I can just select whatever I require and then download. File that I've downloaded, it is a CSV file, so you could use an Excel to open it or you could use any other software available. We can see the information that we have requested for, the accession ID, the release date, the geolocation, as well as the collection date. Now we could use this for any research purpose that we require to do. The NCBI virus database also has information about the proteins, which can also be accessed in the same way as I mentioned. Most of the filters are the same, but currently we'll not be looking at this too much into detail. So I hope that this lecture has given you a better idea of how NCBI can be used to look at nucleotide and genome information for SARS-CoV-2. Thank you, and don't forget to tune in for our next sessions.